Classification of lipids include the simple lipids, ester or fatty acids with various alcohols. Then comes the fats or triglycerides. These are the esters or fatty acids with glycerols. Oil are fats in liquid state. Waxes. Then comes waxes. These are complex. These are the esters or fatty acids with high molecular weight monohydric alcohols. We have already studied a few of the structures in earlier slides as well, and we will also discuss these next lectures as well. Classification is further, they are classified into complex lipids, esters of fatty acids containing groups in addition to an alcohol and fatty acid like phospholipids, glycolipids, or other complex lipids. We have already studied the structures in the earlier lectures, like phosphoric acid residues are there, nitrogen containing bases are the substituents, and they are called glycerophospholipids and sphingospholipids in case when the alcohol is sphingosine. Glycolipids, lipids contain fatty acids, sphingosine, and carbohydrate. From there, the word glyco is coming in this lipid structure. Other complex structures, they are sulfolipids, aminolipids, etc. Precursors and derived lipids. These include fatty acids, glycerols, steroids, other alcohols, fatty aldehyde. They are based on the modification of basic lipids. These all bodies or metabolites, they are resulted from the diabolization of the basic lipids. Now we will talk about the fatty acids. They are the aliphatic carboxylic acids. Have the sugar formula of this aliphatic chain and then the carboxyl at the end. We have already studied their chemistry, the structures in previous lectures. They occur mainly as esters in natural fats and oils, but do occur in the unesterified form as free fatty acids. A transport form found in the plasma. In the plasma, they are transported as free fatty acids. Fatty acids that occur in natural fats are usually straight-chained outlets containing an even number of carbon atoms. The chain may be saturated, containing no double bond, or unsaturated, having more than one or one double bond. According to nature of hydrophobic chain, fatty acids can be classified. They can be saturated, unsaturated, branch chain fatty acids, substituted fatty acids. Saturated fatty acids don't contain double bond, while others they have. Saturated fatty acids may be envisaged as based on acetic acid as the first member of the series, in which CH2 is progressively added between the terminal CH3 and COH group. Like you can join many CH2s. At one terminal there is CH3 and the other end there is the carboxyl group. Fatty acids in biological systems usually contain an even number of carbon atoms, typically between 14 and 24. The 16 and 18 carbon fatty acids are most common. The hydrocarbon chain is almost invariably unbranched in animal fatty acid. A few branched chain fatty acids have also been isolated from both plant and animal sources. These are a few of the examples of saturated fatty acids. You can see based on the number of carbon atoms. Their common name, systematic name, and formulas are there. You would suggest you remember the names, number of carbon atoms, and formulas of the fatty acids with carbon number 10 and higher. They are always even carbon containing fatty acids. Unsaturated fatty acids may further be divided as monounsaturated, polyunsaturated fatty acids. Number already identifies that they have one or more than one double bonds. The configuration of the double bonds in most saturated unsaturated fatty acids is cis type. The double bonds in polyunsaturated fatty acids are separated by at least one methylene group. They are not consecutive double bonds, they are mostly conjugated double bonds. Types of fatty acids according to number of double bonds. Saturated is at the top, no double bond, and there is unsaturation. Unsaturation means hydrogen can be added with one double bond, and there is polyunsaturation with more than one double bond. There are two double bonds and they are not adjacent double bonds. There is one methylene group in between two carbon double bonds. Nomenclature of fatty acids. How fatty acids are named. The systematic name for a fatty acid is derived from the name of its parent hydrocarbon by the substitution of oic for the final E. Like if the hydrocarbon is butane, then instead of E at the end, it will be butanoic acid. For example, the C18 saturated fatty acid is called octadecanoic acid. Octa is 8, deca is 10, it becomes 18, C18. Instead of octadecane, it is written as octadecanoic acid. A C18 fatty acid with one double bond is called octadecanoic acid with two double bonds, and with two double bonds is called octadecadienoic acid, and with three double bonds octadecadienoic acid. The notation 18-0 denotes a C18 fatty acid with no double bonds, whereas 18-2 signifies that there are two double bonds. That's the way how the nomenclature for fatty acids is represented. Carbon atoms are numbered from the, and now comes the issue becomes the issue of the from where the carbon double bonds are starting and how to number the carbon atoms. Carbon atoms are numbered from the carboxylic group, that is the carbon number 1. The carbon atoms adjacent to the carboxylic carbon numbers 2, 3, and 4, also known as alpha, beta, and gamma carbons respectively, and the terminal methyl carbon is known as omega or N carbon. The position I will show you in this one. Here comes this carbon. The terminal CH3 methyl group is omega carbon, and all in between carbons are N carbons, and adjacent to carboxylic group, the first carbon in the carboxylic group is the first carbon. Next to the carboxylic group becomes the alpha, and then beta, and then gamma, so on. The last terminal carbon is the omega carbon. Carbon atoms are numbered from the carboxylic group, and the 2, 3, 4, and the terminal carbon, methyl carbon, is known as omega or N carbon. The position of a double bond is represented by the symbol, that's one of the superscript numbers in the form of a triangle. For example, this sign at 9 indicates a double bond between carbons 9 and 10 of the fatty acid. If there is a symbol of a triangle, then at any number, then it indicates the carbon double bond position. Alternatively, the position of the double bond can be denoted by counting from the distal end, with the omega carbon atom, the methyl carbon, as number 1. That's also the possibility. Omega 9 indicates a double bond, omega 9, from where the counting of the carbon atom is starting from the methyl group, and the 9 omega-9 indicates a double bond on the 9th carbon counting from the omega carbon. In animals, additional double bonds are introduced only between the existing double bond 9603 and the carboxylic carbon, leading to 3 series of fatty acids known as omega-9, omega-6 and omega-3 families respectively. Such kind of fatty acids, they are extracted from animals. Unsaturated fatty acids, their common names are there, family is there, so omega-9, omega-7 or omega-6, and number of carbon atoms, and systematic names are there. You need to draw the structures of the few, and based on the location of the double bonds, you should write the structure, and you should write the this symbolic this type of representation for the fatty acids, for unsaturated fatty acids. These are again a few others, few other examples. Again, few other examples. Yeah, now omega-3 and omega-6 fatty acids. We must have heard about these omega-3 and 6 fatty acids. Omega term is coming from when numbering starts from CH3N. You can see at the right side there is the CH3N, that is methyl, and at the carbon number 3 there is the double bond, then comes at 6 and then comes at 9. So this is the structure for alpha linoleic acid and C18. This is carbon 18, and there are 3 double bonds, and then it is called omega-3 carbon. Then comes icosapentaenoic acid. This is abbreviation is EPA, and this is C20, and that's containing 5 alternate conjugated double bonds, and this is again omega-3. This structure is again omega-3. That is docosahexaenoic acid. Hexanoic is DHA, and you can remember a few of the structures and the number of carbon atoms and their omega-3 and omega-6 characteristics. Cis and trans isomers in unsaturated fatty acids. We will study this lecture in the next presentation.